With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hi everyone, our question says the different steps of recombinant DNA technology are given below randomly. 1. Isolation of DNA fragments or genes to be cloned. 2. Introduction of recombinant DNA into a suitable cell called host. 3. Multiplication or expression of the introduced gene in the host. 4th, selection of the transformed host cells and identification of the clone containing the desired gene or DNA fragment. 5th, insertion of the isolated gene in a suitable plasmid vector. Which of the following represents the correct sequence of steps? So in this question, we have been given the steps involved in recombinant DNA technology. Now these steps have been given randomly, so we have to select the correct option according to the correct sequences of these steps. So as the question is talking about recombinant DNA technology, so let us look at what are the steps involved in recombinant DNA. So the first step is to isolate the piece of DNA that we want to make a recombinant. So the first step is isolate DNA. So we can see here that number 1 will be the first step. Now the second step will be to introduce this DNA into a suitable vector. So we can see that number 5 here will be the second step involved. So this will be the second step. Now after this, after introducing into the vector, the recombinant DNA is now prepared, and the third step will be to introduce this recombinant DNA into a host. So introduce recombinant DNA, this recombinant DNA into any host. So this will be the third step. So we can see here that number 2, that is introduction of recombinant DNA into a suitable cell, usually E. coli. So this will be the third step. Now the fourth step will be selection of transformed cell. So it is very important to select whether the cells have been transformed or not. So this will be the selection process. This step ensures that we are working with only the recombinant organisms and we are not using the non-recombinant organisms. So fourth step will be the fourth step. Now the last step, will of course be multiplication. So now we will make sure that the recombinant gene multiplies. So it will be multiplication so that the recombinant gene or the desired piece of DNA can be expressed. So step 3 will be our last step, 5. So this will be number 5. So we can see here that the correct match here is option C. So these are the correct sequences of the steps involved. So our answer will be the correct answer is option C. Option C. So we can see here that option C provides us all the correct sequence of the steps given here. So this is the answer. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.